second kind of application. Here are two of the heroines of biogeochemistry. This is Antje Bootjes, who is the woman who figured out, or who elucidated and proved the pathway by which methane is oxidized anaerobically in marine sediments. She was at the time at the Max Planck Institute for Marine Microbiology in Bremen. I took this picture when Antje had just been made a fellow of the American Geophysical Union. So there was a happy party. And this is Ann Pearson, who was one of Tim Eglinton's grad students at Woods Hole. And I'm going to be talking about her work, which is why I'm interposing this picture here. Ann's now professor at Harvard. Antje is professor at the University of Bremen, co-director of a major group at the Alfred Wegener Institute in Bremerhaven, and a member of the staff of the Max Planck Institute in Bremen. One of Ann's studies was on materials from the Santa Barbara Basin and the Santa Monica Basin, three cores. SBB is Santa Barbara Basin, Santa Monica Basin, et cetera, off the coast. I mean, these cores are literally off the ends of the runway at LAX. You're sitting there collecting or pulling up core material and jets are going over. But it's a very interesting oceanographic situation. Ann studied three sets of molecular biomarkers from those sediments. This is just plain old cholesterol, which was taken as representative of heterotrophic organisms. This is a 24-methylsterol produced by phytoplankton, so it's a producer signal. And these C40 isoprenoid lipids are distinct products of archaeal organisms that are present in the marine water column. At low abundance, high in the water column, but still there. At greater abundances, lower in the water column. And these products are utterly unique, and so they can be isolated rather cleanly, and they show up nicely in marine sediments. These are isotopic compositions of those products from a depth of about 10 centimeters in the various cores. And you see that the producer is lighter, the heterotroph has got a bit of a trophic enrichment, and the archaea have values apparently characteristic of their different pathway of carbon assimilation. And as we looked at these stable carbon isotopic signals, we just said, oh, well, the archaea are different. They have a different pathway of carbon fixation. They're isotopically enriched, so that's that. Here are the delta 14C signals. And this looks right. The producer and the thing eating the products are equal, just as I showed you they should be. You know, this is a normalized signal, so they both got the same carbon source, they've both got the same delta 14C value, but the archaea looks strange. They're not in the same ballpark, which even if you just look at this all by itself is puzzling. And if you look at the post-bomb samples, it becomes a fascinating mystery in that the delta 13C values, unaffected by the bomb signal, and the delta 14C values for the creatures living up in the photic zone, they shift as you expect. The archaea are pretty much unperturbed. They don't move. So this tells you that the archaea are simply not part. I mean, you could already guess that from the pre-bomb signals, but this utterly nails it. The archaea are not part of the food chain that starts in the photic zone. I contend you never could have known this by any means other than using carbon-14. It's an utterly and marvelously shocking result. So, what are your choices? Well, there's some data you can use. This is carbon-14 available in the records were carbon-14 delta 14C values for dissolved inorganic carbon pre-bomb and post-bomb, and there's dissolved organic carbon also post-bomb. If you look at where the archaea would be able to use, if they were growing autotrophically, a carbon source with the necessary delta 14C value, you can conclude that the archaea were growing as autotrophs at this depth, because here's where they were pre-bomb and here's where they are post-bomb, just slightly enriched. And in practically the only possible interpretation of the isotopic data is that they're growing autotrophically at this depth, and which has turned out to be sustained by microbiological investigations of these organisms. A hypothesis that has to be disposed of is the possibility that they're growing on dissolved organic carbon. There are a bunch of problems with it. Dissolved organic carbon is indeed older, so that could account for the different delta 14C, but the age structure doesn't really fit. The delta 14C of dissolved organic carbon is around minus 460. The value we're seeing is this value around minus 100. So you would have to contend, first of all, that the archaea were checking the delta 14C value before they ate any DOC, and they were selecting this bit right in here. Not very likely. Even if they were doing that, you'd have to say, well, so if they were eating total DOC and just they were eating some old DOC and other modern DOC, you can compute, what the fresh to refractory mixture would have been now and pre-bomb, and it would have to change, which is also, I think, completely unlikely. So the interpretation of autotrophy is the one that stands. Any questions about that as an example of carbon-14 studies? Oh yeah, sorry. I love these illustrations. These are two cartoons describing the distribution of carbon-14 pre and post-bomb. Anne made these for her PhD thesis exam, and I saw them and asked for copies. I think they're just marvelous graphics. So here's the carbon-14 scale. in terms of this delta 14C value. And it tells us what we know. I mean, pre-bomb, carbon-14 is made in the atmosphere, so the atmosphere gets the most intense color. And there's less carbon-14 as you go down. There is some utterly dead carbon in organic materials eroding from ancient rocks on the continents. As you go down in soils, the carbon gets older and older. As you look at sediments, they carry an older signal. It's an average that makes it older. The dissolved organic carbon, some of it's quite a bit older, as we saw, minus 400. But this is the range that you see pre-bomb. Here's post-bomb. Very, very hot atmosphere, hot in terms of the carbon-14. The whole terrestrial biosphere promptly labeled because it's using carbon-14 from the atmosphere. Surface ocean waters, hot compared to the mid-waters because they're pulling in CO2, bomb 14C from the atmosphere. And in the sediments, the deeper sediments are still reflecting the pre-bomb, but the surface sediments are starting to pull down because you've got organic material falling through that water column. They're starting to pull down bomb carbon-14 into the surface sediments. And the comparison of these two, it seems to me, dramatizes the whole bomb pulse, a bomb spike signal, and it's the opportunities that it presents for exploring dynamics. Last bit of dynamics I want to talk about, take us back to Tim Eglinton, who has been investigating this whole sort of transport of carbon from the atmosphere into terrestrial plants, and then this vascular plant litter from that develops on the continents is transported through various pathways to marine sediments. And terrestrial production will, if it gets buried and really built into the geological carbon cycle, it's by transport to marine sediments. And so there are very fascinating questions about the time scale and about the efficiency of these processes that are pulling carbon through all of those steps into the marine sediments. And the theme of Tim's studies in his later years at Woods Hole, and especially now, he's director of one of the institutes at ETH in Zurich, has been a really stunningly broad study of this whole system of transport. And he's produced by now, I think, tens of thousands of measurements of carbon-14 in various organic pools. It's a bewildering, I put into the reading one of the most recent papers, not because it's most assimilable, but because it's most recent, and it gives you, it's a leading reference to a huge body of additional work. And it's a marvelous indication of the state to which these kinds of investigations have advanced. In contrast, this paper I'm showing you right here is one of the earlier simple ones that I can exploit as an example and a demonstration of the motivations for his work. This is about radiocarbon constraints on sources and efficiencies of transport of organic carbon in the Kolyma River, which I'll show you its location. It drains into the East Siberian Sea. Why is this particularly interesting? Well, half of the global soil organic carbon is in Arctic permafrost. 25% of that is in the so-called Yadoma, which is this permafrost dirt that has in it organic material. It's frozen to a depth of 500 meters, and only about 20 centimeters is active in the northern reaches of Siberia. This is, of course, exactly the part of the globe which is now warming most rapidly. And the question is, what's going to happen to this stuff? How rapidly will it be dumped into the ocean, and what will its fate be as it's released? So this is what Tim has been, with a marvelous collection of collaborators, exploring.